the screen that I'm sharing? Yeah, okay, great. Okay, so in the last class we started from fermentation. So just a quick summary. Devangi, would you like to do a quick summary? Are you there, Devangi?
Yes, sir.
Yeah, thank you.
So fermentation is the process of incomplete oxidation of glucose under oxygen deprived or anaerobic conditions.
Correct.
In fermentation, pyruvate is converted into carbon dioxide and ethanol, C2H5OH, and if complete oxidative breakdown takes place, then only carbon dioxide will be produced as a carbon output molecule.
Perfect.
Uh, so, when glucose is converted into pyruvate through
Mm-hmm.
the process of glycolysis, so then the pyruvate gets converted into carbon dioxide and it is known as oxidative breakdown.
Right.
So
But
we
but in case of fermentation, all of the pyruvate does not get converted into carbon dioxide,
yes,
right?
so we have two carbon dioxide plus two
Ethanol.
ethanol.
So total 6 carbon that we started with in glucose, due to fermentation, gets divided into carbon dioxide and ethanol. So which means complete oxidation has not happened, correct? And then we talked about this scheme. What does this tell you? Devangi?
So could you please tell this one?
Yes, I will. So this is simple. So I started with glucose, and through glycolysis, it makes pyruvic acid. So by this time, all of this must be set in your head, right? So glucose is a 6 carbon molecule with which we begin the system, the respiration. It breaks down into two molecules of pyruvate, which is a 3 carbon molecule. And then this pyruvate has, under less oxygen or oxygen deprived condition, can show no oxidation or partial or incomplete oxidation, and can show, if there is no oxidation, then or very less oxidation, so removal of addition of hydrogen, which means hydrogenation, then lactic acid is formed from this pyruvic acid with the enzyme called lactate dehydrogenase. This is, just remember this name. Under partial or incomplete oxidation, as we know, ethanol plus CO2 is formed, right? When very less oxygen is available. So these are the two major pathways of anaerobic respiration, which happens in the absence or scarcity of oxygen, right? We realize that there is no energy production happening here, correct? So whatever energy was produced from glycolysis, which is 2 ATPs, is all that we have produced till now. Okay. Now we go for aerobic respiration next. Would you like to take on, Devangi?
Yes, sir. So, the process that leads to complete oxidation of organic substances like glucose in the presence of oxygen to release carbon dioxide, water, and large amount of energy is called aerobic respiration.
Yes.
And aerobic respiration takes place in the mitochondria. So, pyruvate is transported from cytoplasm into mitochondria.
Yes.
Formula of pyruvate is C3H4O3, and there are two steps. First of all, the complete oxidation of pyruvate takes place by removing all hydrogens that leads to the formation of 3 molecules of carbon dioxide. And this takes place in the mitochondrial matrix. Then the electrons are removed as a part of hydrogen uh, is passed on to oxygen molecule. That is reduction of oxygen and forms water, and ATP is synthesized. And it
Perfect.
is called electron transport chain, and it happens in the inner membrane of mitochondria.
Perfect. So this is the overall reaction that we discussed, right?
Yes,
In
sir.
yeah. So this is for aerobic. Now we have pyruvic acid plus coenzyme A and NAD plus. In the presence of the enzyme pyruvate dehydrogenase and its cofactor magnesium, it forms acetyl CoA, CO2, and NADH plus H plus. Now, as you know, pyruvate is a 3 carbon molecule, acetyl CoA is a 2 carbon molecule, so one molecule of carbon is out as CO2. So we are left with this acetyl CoA. Now, I told you in the last class, why is the cell doing all of this business? Because if you realize, no energy has been formed till now, right? We are just taking pyruvic acid and trying to do something with it. So today we will be starting with why we are doing all of this. Okay, so we will be doing TCA cycle and electron transport chain. So Iram has also joined. Good evening, Iram. How are you doing? Do you have any doubt till here?
Good evening, sir. Yeah, I'm good.
Yeah, any doubts till here?
No.
Okay, so let's see what's happening to this pyruvate and why the acetyl CoA is made. Okay, so I will be starting today with TCA cycle, also known as tricarboxylic acid cycle or citric acid cycle or Krebs cycle, right? Now, what I will be showing in yellow is not a part of the cycle, but just to put a context again. So let's start with pyruvate, because now we have glycolysis is done, we are left with pyruvate, 3 carbon molecule, and then this pyruvate, as I told you, forms acetyl coenzyme, right? Is it clear? Acetyl coenzyme A. In this process, what happened was what went inside of this process is CoA, and what came outside of this process was one CO2 molecule, correct? Till here. Now this acetyl CoA, which is a 2 carbon molecule, 1 second, yes, it is a 2 carbon molecule, now will enter into what is known as the Krebs cycle. So when this enter into Krebs cycle, what I'll be drawing with blue. So Krebs cycle basically starts with the acceptor of acetyl CoA and acetyl CoA itself. Now, acetyl CoA is a 2 carbon molecule, and we know that the first stable product of this cycle is, what is the first stable product of this cycle?
Citric acid.
Yes, it is citric acid. And you must be knowing citric acid is a organic acid, and how many carbons are there in citric acid? 6. It is a 6 carbon molecule. Now look here, a 2 carbon molecule came in and became 6 carbon. So clearly something is accepting this molecule, correct? So the molecule that accepts acetyl CoA must be how many carbons long? Logically, 2 plus dash is equal to 6, a very simple mathematics.
4.
4. So it is basically a 4 carbon molecule called OAA. You have studied about it when I talked about Calvin cycle, it's called oxaloacetic acid. Remember, in plants there is a oxaloacetic acid, right? C3C4 plants. In which plant the first stable product was oxaloacetic acid? C3 or C4?
C4.
Perfect. So in C4 plants there is oxaloacetic acid as a part of their photosynthetic pathway, and from there this oxaloacetic acid is a 4-carbon molecule. It is also the acceptor for acetyl CoA. That's why cell was making acetyl CoA through all of this, okay, without making any energy. So this is the preparation. Now citric acid, so what we'll be doing is, with green again I will be writing the name of the enzyme. So what's happening at this stage is called condensation. Okay, so write down. The first step is condensation of acetyl group with OAA. Condensation of acetyl group with OAA to form citric acid. Any acid, so pyruvic acid is called pyruvate. Oxaloacetic acid can be called oxaloacetate. So I can also call citric acid as inside the cell as citrate. Okay, can I call it citrate? Perfect. And the enzyme which will help in the formation of citric acid from acetyl CoA and OAA, what can you guess the name? It is making citrate. So the name of the enzyme is citrate synthase. It synthesizes citrate. So I'm writing in green the name of the enzyme, okay, for every step. Citrate synthase. You just have to know the enzyme name for entrances, not for your boards. So citrate synthase makes citric acid or citrate. Okay. Now in the next step, this citric acid, the 6-carbon molecule, goes on to form. So there are steps that are not part of your NCRT, but I think you should still know it. Okay. There is a step in between where it forms isocitrate. Okay, it is just citrate only. It is still a 6-carbon molecule. Okay, but just a little bit rearranged. Cool. And then from isocitrate, it forms a very important molecule called alpha ketoglutarate, alpha Kg. This is an important molecule. Why is this an important molecule? Because it is a 5-carbon molecule. Okay, it is a 5-carbon molecule. Isocitrate is 6-carbon. So what happened in between? Something should go out. What should go out? If from 6-carbon you are making a 5-carbon, what should go out?
Carbon dioxide.
Yes, CO2 should go out. So what goes out is CO2 at this step. And very important is you have NAD that comes in and NADH plus H plus that goes out. Now keep remembering this molecule NADH. Okay. Now by this time, if you are already frustrated that what is this happening, where is ATP? If you are waiting for ATP, because we are not making energies, right? The whole point was doing aerobic respiration was to be a complex organism so that we can make more ATPs. But do you see any ATP making? No. So it's obviously, if you are getting frustrated, the frustration is obvious. But I'm just building up because at the end you will realize. So we have till now not make any ATP. and we have made alpha ketoglutarate, which is a 5-carbon molecule. Is it clear? Now from alpha ketoglutarate, again, there are a few steps, but what we will be talking about is succinic acid. There is a succinyl group in between, but we'll not talk about that. Many things happen in between, but what is important step, let me make this a little bigger, is from alpha ketoglutarate we come to succinic acid or succinate. All the acids inside the cell can be written as the form of 8. Remember, alpha ketogluteric acid or alpha ketoglutarate, isocitric acid, isocitrate, citric acid, citrate, oxaloacetic acid, oxaloacetate, pyruvic acid, pyruvate, succinic acid, succinate. Whatever right is correct, not wrong. Now, important thing is it is a 4-carbon molecule. So you can tell me what has happened again. What happened? What should go out?
CO2.
Perfect. One more CO2 release. So you're losing carbon, but you're not making energy. Now what's happening? Now what's happening here is, again, what goes in is NAD plus, and what goes out of this cycle is NADH plus H plus. Again, remember, the cell is planning something molecularly. We are not understanding what's happening right now, but just remember that you are not making energy, but you are making these. You are removing CO2 and making these NADH. But at this step also, just to let you go of your frustration, ATP is formed, but not directly. So there is a GDP. Okay, ATGC, there are 4 bases. So ATP is adenosine triphosphate, and GTP is guanosine triphosphate. Now GTP is formed, but GTP is not ATP, right? So GDP comes in and makes GTP. But don't be disheartened. This GTP one and the most frequent and energy currency is ATP. GTP is also used in plants. So because we are studying respiration in plants, you will find GTP also coming up at this step. Make a hashtag and slash and write ATP comes, okay? Because it does come. So 1 ATP finally, okay? So feel a little better. But the enzyme which makes succinic acid from alpha ketoglutarate. So what's happening here? So there are 2 enzymes. So as I told you, it's a two-step process. So from alpha ketoglutarate, I'm not telling you about the enzyme which makes isocitrate to alpha ketoglutarate because that's not an important step for you. But from alpha ketoglutarate, first there is an enzyme which is called alpha ketoglutarate dehydrogenase. Alpha ketoglutarate dehydrogenase. This comes and take hydrogen. Can you see hydrogen going out from the molecule because of this? Right? Perfect. So everything should make sense what's happening here. And then when this goes out to form succinic acid, and the enzyme which comes is called succinyl CoA synthetase. Okay, this is enzyme to make succinic acid. Now from succinic acid, what forms is your fumarate and malate, which means fumaric acid and malic acid. but I'm just writing directly malic acid. Okay, or let me write it because sometimes this question has come. So from succinic acid, first is fumarate or fumaric acid, and then forms malate or malic acid. Okay, now fumarate and malate both are 4 carbon molecules, just like succinic acid. It does not go beyond 4 carbon, does not reduces, because ultimately from malate you have to reach back to oxaloacetate, which is a 4 carbon. But what is the difference in all these, all 4 carbon molecules, is what comes in and what goes out. So remember, in this step between succinic acid and malate, okay, Fa you see there's a
So-
molecule called, yes.
So can
Yes,
you
Hiram.
explain this again, how malate forms?
How malate forms? Yeah, so basically you see from succinic acid onwards, it is a 4 carbon molecule till oxaloacetate, right? So no removal of carbon happening, only 2 carbon were removed in the form of CO2 here and here, correct, to first make citric acid into alpha ketoglutarate, which is 5 carbon, and then succinic acid, which is 4 carbon. Now this fumarate step is not very important according to NCERT, but I think it is important because of what I'm going to explain here. So from succinic acid forms fumaric acid, okay, also known as fumarate. And from fumarate forms malate. There is only one L here, by the way. MALATE, malate, malic acid, okay. And from malic acid forms oxaloacetic acid. Is it clear?
Yeah.
Now from succinic acid to fumaric acid, there is a molecule called there is a molecule called FAD, not NAD this time, FAD, which converts into FADH2. So also remember this molecule. Again, you must be thinking one ATP you formed, and then again you are doing this nonsense FAD something something business. But just hold on, okay? And so what's happening here is then malate forms. From malate to oxaloacetate, again cell is planning something. Another NAD plus comes in and gets reduced to NADH plus H plus, okay. And then finally you form oxaloacetate back because it's a cycle. So this should continue. Again, this oxaloacetate will trap is succinate. From here you can see that hydrogen is being removed, right, from succinic acid. I'm so sorry, excuse me. From succinic acid or from succinate, can you see that hydrogen is removed? FAD came and FADH2 went, so 2 hydrogen it removed, took with it. So at this step there is an enzyme called succinate dehydrogenase, because it removes hydrogen from succinic acid. And at this step there is an enzyme called fumarase, because it acts on fumarate and then malate. And then again you can see what is happening on malate. Again hydrogen is being removed. So what enzyme will be here? I think you can easily guess the name here. Can you guess the name of enzyme here? Hydrogen is being removed from malate. Is it clear? Okay. So all the enzymes and all the pathways are clear. Now let me pay attention. Let me use some highlighting stuff to highlight something. What do you see here? NADH plus. What do you see here? NADH plus H plus. What do you see here? NADH plus H plus, correct? Let me also use blue. And what do you see here? FADH2, correct? And then just one ATP at the end at this step. And that also not ATP directly, it is a GTP, which through a parallel reaction makes ATP. Is that correct? Is this cycle making sense, everyone? So right now, let me, yes,
What's the full form for GTP?
What's the full form of GTP? Guanosine triphosphate. Adenosine triphosphate, guanosine triphosphate. Okay. GTP is guanosine, GUANOSINE, guanosine. You know ATGC, the bases in the DNA? Adenine, guanine, thymine, cytosine.
Yes.
The same bases are there, okay? ATP is adenosine, where adenine is there. Adenosine is there, sorry. And GTP is guanosine, CTP is cytosine, and TTP is thymine. So GTP and ATP is the most common both in animal and plant system. GTP is present in plant system, not very common in animal system. But I will tell you. So let's write the steps first. So this is the first step. This is the second step. Let's say I'm according to NCERT. Okay, let's say this is the third step. Then this is the fourth step, fifth step, and sixth step. I told you about sixth step. There are more steps, but not relevant to you. Okay. So first step is called a condensation step, where acetyl CoA condenses with oxaloacetic acid. So write down step 1 is condensation step. Then step 2, 3. So step 2 and 3 are called decarboxylation step. One is condensation step, step 2 and 3 to form succinic acid. Finally, till succinic acid, step 2 and 3 is called decarboxylation, because you can understand why it is called decarboxylation, because carbon dioxide is removed, right? Is it clear? After that, rest of the steps from succinic acid to oxaloacetic acid are oxidation steps. Why it is oxidation steps? Because at every step there is hydrogen which is being removed. Oxygen is not being added, but hydrogen is being removed. And if you remember from your chemistry classes, even removal of hydrogen is called oxidation, right? Is it clear, everyone?
Sir,
Yes, Devangi.
FADH2 provides energy.
I will talk about it. Just remember these names, NADH, FADH2, because these are the major players. There was just one GTP produced. So why is cell doing all of this? We'll talk about it. But let's go one step at a time. So is this clear? Step 1 is condensation, 2 and 3 are decarboxylation, 4, 5 and 6 are oxidation. Is it clear?
Yes, sir.
Okay, now based on this, okay, also write one more point about GTP. Write down the GTP which is produced, after step 3, the GTP which is produced, the only GTP which is produced in this cycle, okay, in a coupled reaction, okay, in a different reaction, leads to synthesis of ATP, So we can take it as a ATP production. We can take this GTP as a proxy to this ATP. Okay, is that clear? Now let's try to break down this very complex reaction into parts. So what we started with this in this cycle is again pyruvic acid, correct? And what all things came to react? I saw a lot of NADs coming in, NAD plus coming and reacting. I also see FAD coming and reacting. I also see water. Okay. So water came at 2 steps, which I have not mentioned, but just remember that water came. And one ADP also here. Okay. This GDP can be taken a proxy for a ADP. Okay. Because I told you that in a parallel reaction, this GTP goes down to make a ATP. So we say that this GTP leads to an ATP, so we will count it as ATP. So ADP plus a PI. So all of this is happening in, where is it happening? Mitochondria. And where in mitochondria? Mitochondrial matrix. And what all things are we getting out of it? We got CO2 plus we got lots of NADH. How many? We will count. We got H plus. We also got some FADH2. And we got, finally at the end, we got one ATP. So let's count how many pyruvic acid we started with in this cycle. 1, right? How many NAD plus were used? 4. Can you count? 1, 2, 3. There's the 4th one. For the 4th step, I have not told you. Anyways, so just remember 4. 1 FAD at this step, which is clear. And remember 2 H2, 2 water molecules were used, 1 ATP was used. We got out 3 CO2, 4 NADH, 4 H plus, 1 FADH2 and 1 ATP. This is the overall reaction till here. We have come a long way. We started with glucose, did glycolysis, then started with the first aerobic part, which happens in the mitochondrial matrix. Okay. Is it clear till here? So if I ask a question based on whatever has happened till now, till here, let's do some calculation. So per glucose, how many ATP we got till here? Per glucose, how many ATPs we will get through TCA? 2 ATPs. Is it clear? Is it clear, everyone? Because one pyruvate gives one ATP, so one glucose gives two pyruvate. So this cycle will run 2 times because of one glucose. So 2 pyruvate will give 2 ATPs. So 2 ATPs are produced in TCA per glucose. Doesn't look like a very good cycle, right? Because two ATPs we also got from glycolysis, a simple glycolysis where no mitochondria was required. So till now, you must be feeling a little disappointed and thinking that what is the point of respiration, aerobic respiration at all? We have not seen any oxygen coming into the picture till now. Okay, we have not seen any large formation of ATP till now. I already gave you a spoiler in the beginning that there will be around 36 to 38 ATPs formed in aerobic respiration. Till now, we have only formed 4, 2 in glycolysis, 2 in TCA, right? But that's the point. Picture abhi baki hai. So the main thing for which the cell was preparing all this time were these molecules. These 4 molecules of NADH and this one molecule of FADH2 per pyruvate. So per glucose, tell me, per glucose, how many molecules of NADH will be there? 8 molecules of NADH will be there, right? And 2 molecules of FADH2 will be there. Is that correct? NADH plus H plus, let me write. Is that correct, everyone? Everyone is on the same page till here. This is a little convoluted, but do you understand this thing till TCA cycle? Hiram Janya? Perfect. Now, these molecules, so write down, let's write something. Have you written this? That till now, till TCA per glucose, we get 2 ATP in TCA and 8 NADH and 2 FADH2. Everyone has written that? And everyone has copied this cycle? Or do you want me to stop at this so you can copy it? Those who have copied, give me a thumbs up so that I can move forward. And anyone who needs time, please write in the chat you need time or tell me. Do you need time, people?
Yes, sir, 2 minutes.
Yes, take time. Okay, I'll come back in 2 minutes. I'll get some water, okay? Is that okay? Devangi, Hiram? Okay, done, everyone?
Yes.
Okay, so let's move forward. Okay, so till now we realize that the cell has managed to produce a lot of these molecules called NADH, FADH2, but very less of ATPs. But the idea is, because this is, remember, the first part. So aerobic respiration happens in two parts. One thing which is happening in the mitochondrial matrix is that all hydrogens removed from pyruvate and 3 molecules of CO2 produced. So we realized, where are those 3 carbons removed? So one is removed here, second is removed here, and third is removed here, right? And lots of hydrogen is removed at every step. One hydrogen is removed here, one hydrogen is removed here, one hydrogen is removed here, and one hydrogen is removed here. Oh, I forgot to tell you that the one step where I did not tell where the NAD comes is this step. So at this step, NAD plus comes and leaves as NADH plus H plus. Okay, so you can finally realize that lots of hydrogen is taken from pyruvate throughout this cycle, and CO2 is also taken out from this pyruvate at multiple, specifically at 3 places. This is what this reaction that we wrote was, this one. Okay, and this is happening inside the mitochondrial matrix. But the last part, the climax of the movie, is still left, and that is where the inner mitochondrial membrane, this electron transport chain happens. Okay, so let's go and read about electron transport chain. And that is where I promise, and I promise because we know it happens. So I promise that lots of ATP will be formed and finally you will be happy. So electron transport chain. Now, what we are going to read is a very simplified version of electron transport chain. Okay, and the major things to remember, it's also known as electron transport system or ETS. Okay, it's called ETC or ETS. It's also known as the oxidative phosphorylation. Now this is where oxygen will come in. Okay, so the hero molecule will come in at the end, in the last cycle. Till before this, oxygen did not come and play a part in aerobic respiration also. So write down NADH plus H plus. So let's write certain things down. So first thing, in the TCA cycle, NADH plus H plus and FADH2 are high energy molecules. So I think Devangi was asking this question, right?
Yes, sir.
High energy molecule intermediates. So they are called intermediate molecules. Why they are called intermediates? Because they are never used. So they are like check. So if I give you a check of 10 lakh rupees, that check itself will be of no use to you. It is a very high energy paper in your hand, but you cannot use it. You cannot shop with it. You cannot go and buy clothes, foods, you can go and buy a car or something like that. But like, let's talk about basic requirements of a cell and a human. So is it a demand draft or a checkbook? Is it of any use to you in day-to-day life? No, right? You need the currency in use in the paper notes or the coins or digital payment method, etc. So, but having these in hand is also very important because you can convert it. And this is exactly what happens at the last step. So write down these high energy molecules are oxidized through the electron transport system. The electron transport system, and the electrons are passed on to oxygen molecule, resulting in the formation of water. If you reduce oxygen, give all the hydrogen to oxygen, then it will form water, correct? So now do you realize why cell was making NADH and FADH2 in the citric acid cycle and not making ATP directly? It was setting up the stage, it was setting up the baseline, the foundation, by making, instead of going and making chillers, currencies, paper notes, it was making checkbooks and demand drafts, high energy molecules. Because in the next step, in the membrane, inner membrane of the mitochondria, where electron transport chain happens, these high energy molecules can be oxidized and their energy can be used to make ATP, and their electrons can be passed on to oxygen to make water. Make sense? Thumbs up if that is clear. Everyone, Hiram, Jania? Perfect. So now you got to know the plan of the cell, right? Of an aerobic cell. Good. So let's define it. Write down the metabolic pathway through which we are defining electron transport system. Define electron transport system. The metabolic pathway through which the electron passes from one carrier to another. The electron passes from one carrier to another is called electron transport system. Is called electron transport system, and it is present in electron transport system and it is present in the inner mitochondrial membrane. It is present in the inner mitochondrial membrane. Okay. Is it clear, everyone? Now, I will make a diagram and will try to explain you. So pay attention to this. So we all know that mitochondria. Let's start with the mitochondria. So we all know that mitochondria has an outer membrane which is smaller and an inner membrane which is like this, What is bigger, right? Sorry. Yeah, and there is a inner membrane which is like this, correct? Everyone is clear? Now let's look at a big zoom in section of this region, let's say. So if you look at this region, what do you see? You see a inner membrane, right? So let's say this is the inner membrane. I have straightened it. And this membrane will have some thickness, right? This is the inner membrane. And I will make with a highlighter an outer membrane. And let's say, and let's say this is a outer membrane. Is it clear?
Inner membrane would have enfoldings, right?
Yeah, so I've straightened it up, like just to make things easier. I'm straightening it up. Okay. If you want, let's make it like this, a little foldings, not very straight. Is that clear? Now, is it okay? I have taken a very small
Yeah.
part, which is like straightened. Okay. So if this is outer membrane of the mitochondria, and this is inner membrane of the mitochondria, what is this space in between called? Can I call it as the intermembrane space? Right? And this is inside, so this will be the mitochondrial matrix. Is this clear? What are we making here? Everyone? Outer membrane, intermembrane space, inner membrane, and mitochondrial matrix. So this whole thing from here to here is clear, right? Yes, no, something? Perfect. Cool. Now what happens in this inner membrane where these electron transport chain happens? There are certain complexes which are present. So first one is called complex 1. This is complex 1. Then there is a some other color from it. Better. Let's use this color. Then there is another complex called complex 3. Okay, you will think complex 2, but it is like 1, 3, 2, and 4. I know it's a little weird, but that's how it is. Okay, so I'll tell you why it is like that. And then there is a complex called complex 2. And let me use another color. I'm out of colors, but let's say red. And then there is a complex 4. And at the end, we have the molecule for which we all were waiting. Looks like a motor, right? This is the most important thing in this membrane. So let's write first off the names in the order. So the order is from here, okay? Let's say. So the first thing that comes is called complex 1. Okay. Then complex 3, then complex 2, then complex 4, and then this is called ATP synthase. This is the protein that I was talking about. that actually rotates like a motor and fixes ADP and PI into ATP. Now what happens? Let's see. Should I remove some of these markings like this to make it a little clear? I hope you have noted this down, right? Everyone? Noted this? 1, 2, 1, 3, 2, 4 and synthase. Okay, the complex 1, write down the names. Complex 1 is also known as NADH dehydrogenase, known as NADH dehydrogenase. It has this enzyme. So it's like it's not known as, I should not say it's known as, it is oxidized. Okay, NADH dehydrogenase, and due to which what happens through this complex, all the so I'm erasing this name complex 1, and what happens is, remember in the matrix, so this is the matrix, right? So in the matrix there was NADH plus H plus being produced, right? Here a lot of NADH plus H plus was being produced in TCA cycle. Yes or no? So all this NADH plus H plus comes to the membrane, inner membrane, and becomes NAD plus again, so that they can again go back and do the same thing for the cycle. So they are taking hydrogen from these molecules in the citric acid cycle and they are giving it back to complex 1. Okay, this is 1, this is 3, this is 2, this is 4. I'm removing the names from here. And when it does that, what happens to all the hydrogen? All the hydrogen moves through this complex and comes out and accumulates in the inner, or sorry, intermembranous space. Is it clear, everyone? Now do you understand what is happening slowly and slowly? This is the bigger plan of the cell. By this enzyme, you make NADH lose their hydrogen into the intermembrane space and become NAD back and become back into NAD plus, go back to TCA and do what they were doing. And they continuously keep doing it for the rest of your lives. Is it clear? And then complex 3, write down, complex 3 has it has 2 other enzymes and complexes which are a little okay, let's write down this. The electrons are, so you can see that the H plus, which is your proton, is accumulated in the intermembrane space. What happens to the electrons? Write down, the electrons from complex 1 are transferred to, so I'll mark it like this here. So the electrons, remember it was an electron transport chain. So electrons keep going from one complex to another. So write down, the electrons from complex 1 is transferred to ubiquinone. Ubiquinone. So ubiquinone is sitting in the membrane between complex 1 and complex 3. Ubiquinone, spelling is UBIQUINONE. I will write it here. Ubiquinone. Just some details. I know you don't have to remember these details for your boards, but just remember that from complex 1 the electrons go to ubiquinone. Okay. From there the electrons go to complex 3. Okay, and then to complex 2. No. So from 3 it goes to complex 4, not to 2. That's one, because I'll tell you. Oh, okay. So the electron goes to complex 2. Right down. From complex 3 the electron goes to complex 4. So what does complex 2 does? So complex 2 has FADH2. Okay, FADH2 comes in complex 2. And write down next line after ubiquinone, that ubiquinone receives reducing electrons via or from through. Ubiquinone receives reducing electrons through FADH2 in the complex 2. Okay, that is a function of complex 2. So complex 2 kind of gives back to complex 3. So electrons from complex 1 goes to complex 3. There is ubiquinone between 1 and 3. When it goes to complex 3 from complex 2, FADH2 gives reducing equivalents to the same ubiquinone. From 3 it goes to 4. Okay, and in complex 4 it is where this happens. So in complex 4 this reaction happens, that you need O2 plus hydrogen and make it into water. So if you balance the reaction, it is a weird reaction because half O2, which doesn't make sense, plus hydrogen, which is present here, which was being transferred, right? So all the electrons, when it reaches here, it is hydrogen in the form of hydrogen, correct? And half O2 plus 2 hydrogen from this complex 4 makes H2O, water. So this is their final water formation, and the electron transport chain completes. So complex 1 to 3, 3 to 4, and 2 in between helps in the reduction of ubiquinone in complex 3. That's all you have to know. There are a lot of other things happening, but you don't have to know, or you don't have to worry about that. And remember that there are 3 complexes. Just leave complex 2. Complex 2 does not play a part in maintaining the proton gradient. So from all other complexes, proton leaks into, not leaks, but is pumped into the intermembrane space, except for complex 2. Even from here, H plus keeps accumulating. Okay, so complex 1, complex 3, and complex 4 helps in proton gradient accumulation in the intermembrane space. Is it clear? The final one, which is the ATP synthase, we call it the complex 5, just by naming. This is the complex 5, but remember the name, it is called ATP synthase. Now will happen the most important thing. And the thing is that all this proton which was accumulating in this membrane, intermembrane space, okay, so there'll be a high gradient of these protons, correct? We finally formed water and that is done. So all these protons now start going through this ATP synthase one by one, okay, and they come back into the inner side. And when they do this, this whole thing rotates, okay. And here what happens in this rotation, ADP plus PI comes and ATP is formed, lots and lots of ATP. So you have a motor in place to make ATP. How does that sound? Cool, right? Is it clear, people? And this gradient where you make positive ions in the outside of the inner membrane and negative ions, so the inner side of the inner membrane, which is the matrix, so this side, which is the matrix will be negatively charged, right, as compared to the inner intermembrane space, which is the outer side of the inner membrane, that is positively charged. So this kind of gradient is known as electrochemical gradient. Is it clear? This is the most important part of this chapter. So through this process, the cell ends up making the bulk of ATPs, okay, around 34 to 36 ATPs are made through this process. Is that clear, everyone? Because you can actually drive this motor through the hydrogen coming in through the ATP synthase, which is the 5th complex, and ADP and PI comes in into this and ATP goes out. So we finally get a lot of ATP and we finally get water through electron transport chain. Any doubt in this? This is happening in the inner membrane of mitochondria. Please draw this and let me know if there is any doubt. Now please go back and read from NCERT the electron transport chain. So the way NCERT has written is whatever I have explained here, NCERT has written it. And if there is a point which NCERT does not talks about, or a point that NCERT talks about and I have missed here, please let me know in the next class, okay? Like there's nothing important that we have missed for your exams. But if there is a point that you read in NCERT and you could not understand based on our discussion from the class, please bring it up in the next class. So this chapter has some tail ending parts, some theoretical parts like respiratory quotient and balance sheet and amphibolic pathway that we will deal with in the next class in half an hour, first half an hour, and this chapter will be done. Any doubts, Iram and Jania? Chania has no doubt. What about you, Iram?
No, sir.
No. So please read from NCRT to get clarity and get your revise your concepts. Basically, that's what I'm saying. These are a little fact heavy parts of the chapter. Okay. So if you just listen to it as a story in the class and then forget about it, it will be very difficult. It will be very easy to wipe out from your memory.